Good afternoon, everyone, and welcome to the GenQA Focus On webinar on postnatal CNV classification. We have two excellent speakers for you today. The first is Angharad Williams, who soon to become the lead of the developmental disorder section at the Wales Medical Genomics Service in Cardiff. She has a lot of experience in her ACGH for developmental disorders, large NGS panels for epilepsy and intellectual disability, and is involved in the new rapid whole genome sequencing service for severely unwell babies. She also holds Part 1 of the Royal College of Pathologists exam in genetics. The second speaker is Melody Taberner. She's a Deputy Director with GenQA covering the cytogenomic EQAs. She joined GenQA last year, having previously worked in an NHS diagnostic cytogenetics laboratory for 19 years. She's got broad experience in all areas of cytogenetics and is a Fellow of the Royal College of Pathologists in clinical cytogenetics. We also have one of our assessors for the GenQA Pathogenicity of postnatal germline CNV EQA joining us for the Q&A. This is Sian Corrin, who's the lead for the rare diseases for the All Wales Medical Genomics Service. She's got a lot of cytogenomic experience and is a Fellow of the Royal College of Pathologists in clinical cytogenetics. If you've got any questions for the speakers, can I ask that you add them to the question box and we'll address them at the end of the presentation. So I'll now hand you over to Angharad Williams for the first part of the talk.
Thank you. Good afternoon, everyone. I will be doing an overview of investigation of CNVs and classification. Next slide, please. Thank you. So firstly, best practice guidelines. There are a number of best practice guidelines out there that relate to CNV and array analysis in particular. And I've highlighted on this slide some of the important ones that we, for example, use in our laboratory. There's the Association for Clinical Cytogenetics, the postnatal chromosomal microarray best practice guidelines, which were released in 2011. There are the American College of Medical Genetics previous guidelines, also from 2011. And there's the European Guidelines for Constitutional Cytogenomic Analysis in 2019. And of note is that we will often refer to the ACMG best practice and ACGS best practice guidelines for variant classification in rare disease for the single nucleotide variants, because this can help with classification of intragenic imbalances. You'll note that some of these guidelines date back to 2011, so it wasn't until very recently in 2019 that the ACMG released new technical standards for CNV analysis in the constitutional setting. And these are very similar in style to their SNV guidelines, and we will be speaking about those in a little bit. So firstly, the classification categories that we use, we're in line with the SNV guidelines, and we use the 5 classification categories that are benign, likely benign, uncertain significance, and internally we might refer to these as hot and cold. So hot being having more pathogenic information, or cold being maybe more like a benign. And then we have likely pathogenic and pathogenic. So today I'll be going through some of the principles of CNV classification, and this is an overview of some of the main points that I'll be covering. Firstly, before I get into the kind of bioinformatic tools and what kind of websites you can use, I wanted to point out that it's important to bear in mind throughout your analysis whether you're analysing a deletion or duplication, as these have different mechanisms of action and are often recorded differently and separately in the databases I will be talking about. And you usually can't cross-use this evidence. And another thing that I won't go into depth here is to look at recurrent regions in your CNV, as by identifying these, you can often quickly get to a classification. So some of the things I'll be talking about is population data, patients with similar imbalances and phenotypes, inheritance and segregation and family studies, but mostly it'll be about size and gene content of a CNV and looking at evidence for dosage sensitivity. So one of the very first things that we would look at in our analysis is internal laboratory data, and it's actually recommended in the best practice guidelines that labs document all CNVs in an internal database for ease of future classification. And having your own database of CNVs allows for rapid interpretation of variants, especially the more common ones. For example, benign population variants or technical artifacts or recurrent pathogenic CNVs. And it's worth noting that variants may need to be periodically reviewed for new information before reporting based on internal laboratory data. So population data is a vital tool for indicating if a CNV is likely to be benign or not. Some of the main databases for CNVs are DGV, including DGV Gold, and NOMAD SV, and that's NOMAD structural variants. These generally contain CNVs from so-called unaffected populations. For example, the NOMAD SV contains data mainly from adults with no known pediatric disorders. If you are lucky enough to find the same variant, either as a loss or a gain, at more than 1% in one of these databases is very strong evidence that you can call your CNV benign. Great. So size and gene content are going to be a large part of what I talk about, and it will be the basis of your CNV investigation. And it's how you'll try to predict the impact of the CNV in your patient. The size of the imbalance is not always a reliable indicator of pathogenicity. There are large CNVs of over kind of megabases in size in these population databases that have been shown to be benign in general populations. When assessing the gene content of CNV, you're really looking for evidence of disorders with matching disease mechanisms, such as haploinsufficiency in a deletion, and matching phenotype in your patient as well. And there are a number of websites and tools you can use to look at gene content, but I actually prefer to use Decipher. It's a really comprehensive tool that brings in many different websites and analysis packages into one place. And the screenshot at the bottom there is the output of a search, and you can use your genomic coordinates in the search box at the top. And by clicking on the gene tab in the middle there, you get the numbers of genes, but you can also filter these by whether they're OMIM, OMIM morbid, DGG2P, or protein coding, for example. And then the table underneath gives lots of links out to different websites and information and tools, such as OMIM or DGG2P, or the Clingen haplo and triplosensitivity scores, and also gives in silico predictor tools as well. So OMIM is really useful for further information on genes, including gene function, animal studies, any link disorders and their kind of inheritance patterns. And it will also give you some commonly reported variants as well, so it's a good place to start. And by clicking on the kind of phenotype OMIM number that you can see in the table there, if we go to the next slide, you can get disease-specific pages that link out from these genes, and these give really good clinical synopses of the phenotype. That's the orange box at the bottom, but also give overviews of relevant literature, and also will give affected patients and any family testing that was done there, and then the publication links as well. So the Clingen Dosage Sensitivity Map is really important, and it's a huge part of the evidence for the new ACMG CNV guidelines as well. Genes and recurrent CNV regions are curated by the Clingen group, and they will judge if there's enough evidence in the literature for dosage pathogenicity. They will then give a haploinsufficiency or a triplosensitivity score to each gene or region that has been curated as a measure of the evidence available. And this table shows the rating system from 3 being sufficient evidence for dosage pathogenicity, down to 0 being no evidence. And a haplo or triplo score of 3 is considered to be strong evidence, and CNVs can be classified as pathogenic based on this evidence. And just to point out, when using this website, you should check the date that your gene of interest was evaluated on. In some cases, this might be some time ago. Some of them are from 2012. And so a literature review would be needed to find if there's more recent evidence that is applicable. So like with SNVs, in silico tools can be used to predict the likelihood of haploinsufficiency. And they can be used as supporting evidence, though not as strong as the haplo or triplo scores. And you can find these in silico predictors on Decipher, and they're shown in the screenshot here, and also at the gene level in Nomad. And then the red boxes here show the significance of the scores in Decipher. So the PLI score is the probability that a gene is intolerant to loss of function mutations. And the closer the number is to 1, the more probable that the gene is affected by loss of function variants. And then the percentage HI is the probability of being a haploinsufficient gene. And conversely, the lower the value here, the more likely the gene is to be affected by loss of function variants. And you'll notice I'm talking almost exclusively about haploinsufficiency. So what about triplosensitivity? And there are currently very few triplosensitivity predictors. However, there was a paper by Collins in 2021 in the Medical Archives that has attempted to create triplosensitivity scores for genes. And so these are coming in the near future. So patients reported in the literature or on databases that have the same imbalance or similar and a similar phenotype can add pathogenicity evidence to your classification, especially if they've been reported as de novo, for example. And there are many databases with really excellent patient data sets in them. Decipher is one, which has lots of data from many UK NHS labs. And ClinVar, which was previously ISCA, I believe, as well. And you can also look for patients that have been reported in the literature using search engines such as PubMed, and the Human Gene Mutation Database, or HGMD, gives gene level variants. So this can be helpful when looking at kind of smaller CNVs or at the individual gene level. So this is a very quick view of the Decipher genome browser tool, which allows you to search databases with your genomic coordinates, as I said before. And you're able to see individual overlapping or matching Decipher patients, the red and blue lines in the middle there. And you can filter these based on deletion or duplication. And by clicking on these, you can go and look at individual patient profiles, and there might be inheritance information, whether it's been found to be inherited or de novo. And there is sometimes phenotypic information and pictures. And then sometimes also there's a pathogenicity classification as well. And within this gene browser as well, you can access the ClinVar CNV patients. And there's also the Nomad SV, the DGV Gold, there's gene content, and there's also syndrome regions in there as well. So it's a really helpful tool to use. So depending on your testing methods, you may have inheritance information while you're analysing your CNV. For example, from whole genome or whole exome sequencing, you may have run trios, and you'll have that information up front. All this may be collected after reporting. Testing of patients or siblings may help in the interpretation. For example, a de novo variant will add evidence that a CNV is disease-causing, where other family members are not affected, or segregation among affected family members may be possible. Conversely, the CNV may be found in unaffected family members, and so this would be evidence towards your CNV being benign. And you may need to reassess your classification if you've performed family testing after reporting. And this can be especially useful if you have a VUS and the evidence can then change the classification to, for example, pathogenic or likely benign. Family studies are also important for recurrence risks for the parents and the families themselves. And it's really essential that genetic counselling is involved when doing these kinds of studies. So finally, some things to be cautious about when doing CNV classification and interpretation. And this is by no means an exhaustive list. Firstly, so structural information can be really important to interpretation, but you won't often be given it up front if you're doing array or whole genome sequencing, for example. So sometimes further testing, such as FISH or gene banding, might be needed to fully interpret a result. An example for this might be the 15q11, q13 duplication. This can either be in tandem or is sometimes a small marker chromosome, and these can have different interpretations attached to them. A loss of an autosomal recessive gene indicates carrier status, which may not often be reported, but you should still check whether the phenotype associated with those genes is relevant to your patient and whether further sequencing or further testing needs to be done to look for that variant on a second allele. Intragenic CNVs can be assessed using SNV guidelines. They're really helpful, particularly the PVS1 guidelines, which do have instructions for deletions and duplications of single or multiple exons, so it's worth bearing these in mind. And where a CNV has no protein coding genes, it might be tempting to call it benign outright, but there may be non-coding OMIM genes or regulatory regions, so this should be checked before classification. And a good example of this are the downstream regulatory regions associated with the SHOX genes. CNVs on the X can be tricky and they need to be analysed with respect to both male and females, as even though a female may be unaffected due to X inactivation, it may be relevant to male offspring, and this may need to be reported in some cases. A number of recurrent microdeletion and duplication syndrome regions have been reported with reduced penetrance or are neurosusceptibility regions. And so these can be commonly seen in population databases. So it's worth checking whether you have these recurrent regions before using population data alone. And finally, when using array technology in particular, there may be genes in the maximum region of the call that have important clinical implications. So these should be checked, and where needed, an orthogonal method such as qPCR or MLPA could be used to confirm that the gene is within the region of interest and is therefore relevant to your patient. So just finally from me, a little bit on the new ACMG CNV guidelines, as Melody will be speaking about these in her section. The new CNV guidelines were released by the American College of Medical Genetics and Genomics in 2014. And they've introduced a quantitative evidence-based scoring framework that has a kind of calculation effect. And these are to encourage consistency and transparency between labs that are doing CNV analysis. They're very similar in style to the SNV guidelines, and so are useful from that respect. And the ACGS are currently in the process of adapting these guidelines in the UK. So this is an image taken from the guidelines of the beginning of the loss table. There are duplication and deletion tables, so they vary slightly between the two. And the scoring system is essentially trying to weight evidence to come to a final total, which you can then use to assign the classification. The scoring is given at the top of the table there, and more than 0.99 points is classed as pathogenic, whereas less than minus 0.99 points is benign. And VUSs are a huge category in the middle of between 0.89 to minus 0.89 points. And these metrics, or from the ACMG, take some elements from the SNV guidelines, such as the use of PVS1 and the scoring of individuals with de novo variants. The tables follow the 5 main categories, which are in line with some of the topics that I've spoken about today. And finally, there's a link on this page to a helpful calculator online, which Clingen created, which allows you to assign your evidence and give it varying strengths to come to a total and calculate your classification. I think it's now time for me to pass on to Melody.
Thank you. Yes, so we've had an overview of the CNV classification, and now we're going to look at how it's being applied. And I will summarise the results of the GenQA 2020-2021 Pathogenicity of Germline Postnatal CNVs EQA. So this was a pilot EQA which opened in January of 2021. This material has previously been assessed as part of the postnatal CNV EQA, which is a scheme where we send out DNA samples for laboratories to process according to their usual procedures and submit an interpreted report. We also were sending out surveys to assess the classification of CNVs, and that's the part that we've now separated from the CNV EQA into this new pilot EQA, which is purely online. There are no samples sent out for this one. So the recent EQA consisted of 4 case scenarios, and the aims were as follows: to assess the classification of the pathogenicity of copy number variants provided, to assess the classification of the clinical significance of the findings for the patient, and this is in recognition of the uncoupling of classification versus clinical significance, which is now thought to be standard procedure. And we are also assessing the reporting decisions for each CNV provided within the context of the case scenario. So participants were provided with forms to submit their results on, to show their classifications and the justification for those. And we provided either a table which mimicked the American guidelines for the participants to fill in their classifications and scores, or a free text box for those who were using their own justifications. The marking was based on providing the correct classifications for the CNVs and the clinical significance, and making an appropriate reporting decision based on the case. So for this initial pilot EQA, we had 66 laboratories who submitted results, and these were from a wide range of countries. So there were 22 countries involved, and we had most participants from Spain, the United Kingdom, and Italy. So in the next part of the webinar, we'll be summarising the EQA results from this recent EQA. There's a fully interpreted report which has been published for those participants who took part in the 2020-2021 EQA. And this report will also be provided for those who are going to participate in the next round, which will open in the autumn of 2021. Participants also receive individual feedback and their own laboratory scores on an individual laboratory report, which is sent out to them once the assessment process has been completed. So I'm just going to run through the 4 cases which were provided for this EQA now, and explain the justifications for the classifications and any problems that were, or challenges that were encountered. So the first case was a young boy who was referred with mild global developmental delay, especially of language. He had delayed gross motor skills and hypermobility. There was no family history information provided. And the result that was given was a 368 kb terminal loss of the 10p15.3 region. The assessors considered that the expected classification for this CNV would be pathogenic, and also for this particular patient, it should be interpreted as pathogenic. And therefore, the assessors considered that the CNV should definitely be reported. So the justification for this pathogenic CNV classification is that this loss contains the gene ZMYND11, and this is an OMIM morbid dominant disease gene, and several truncating variants have been described in patients with global developmental delay or intellectual disability, just like in this patient. The scoring for this gene is a high haploinsufficient score, so a maximum score for Clingen of 3 and PLI of 1. And ZMYND11 is considered to be the critical gene for the clinical phenotype of the 10p15.3 microdeletion syndrome. So you can clearly see there's a lot of evidence that this CNV is pathogenic. And in terms of any polymorphic variants, there were no losses reported on the database of genomic variation when that was accessed. So there's very little evidence for benign classification. In terms of the clinical significance for this patient, the clinical features described were very consistent with previous reported cases in terms of the developmental delay. However, the hypermobility was not completely explained by this CNV, and therefore the assessors also allowed a likely pathogenic classification. There wasn't any family history provided for this patient, but that would not change the classification, as both de novo and inherited cases of this microdeletion syndrome occur. So the assessors considered that the CNV was pathogenic, it should be reported, and it's consistent with the patient's phenotype. So just under half of the participants applied the American guidelines, and the majority of all of participants had no problem with this case and correctly classified the CNV as pathogenic. And where the guidelines were applied, there was a high level of consistency of scoring for sections 1 and 2, with all participants assigning a maximum score of 1 for section 2A, which means, as Angharad mentioned, the score of over 0.99 would mean it would be classified as pathogenic. Section 3 was correctly scored as 0, and some participants chose to skip this, presumably because the pathogenic status had already been reached. But it's important to note that working through the remaining sections, and particularly section 4, which focuses on literature evaluation, should be considered even when you've already reached pathogenic status, to determine if there's additional information of clinical relevance to report. For the EQA summary report, we did not comment on section 4, as obviously some of this evidence will be obtained from internal laboratory data. And as the inheritance information was unavailable and uninformative, there was a bit of variation for the scoring assigned to section 5. So we're showing the submissions in terms of these bubble plots, where the size of the bubble corresponds to the proportion of participants who assigned that classification, and the colour determines whether the classification was an expected classification in green, an acceptable classification in yellow, or an incorrect classification in red. And those classifications classed as incorrect, those participants received a deduction in their score. So we can see clearly from this bubble plot that there was a high level of consensus, and the majority of participants assigned the expected classifications. There were just 7 participants who received a deduction in this case. And all the participants correctly stated that they would report this CNV, so there were no issues with that. So moving on to the second case, this was a newborn girl who was dysmorphic with a left hyperplastic heart and coarctation of the aorta. Again, the family history was not given, and the reported result was a small 61 kb gain of 22q13.33. The expected classifications were a variant of uncertain significance for both the CNV classification and the clinical significance for the patient. And where variants of uncertain significance are found, laboratories should apply their local policy. Whether to report it will depend largely on current local guidelines and obviously be very specific to each case. So the justification for these variant of uncertain significance classifications are that the gain detected partially overlaps the SHANK3 gene. And this is an OMIM morbid dominant disease gene associated with Phelan-McDermid syndrome. And although there's a lot of evidence for haploinsufficiency of this gene, there's no evidence for triplosensitivity according to Clingen. And it's also unknown whether this partial copy number gain disrupts the coding sequence of SHANK3. The database of genomic variants shows a few overlapping gains. So overall, there's no convincing evidence for pathogenicity and no convincing evidence that this gain is benign. The clinical significance for the patient is also uncertain. The patient's very young, less than 1 month old, and it's possible that not all the phenotypic features are apparent yet or whether they've been described correctly. However, the phenotype could be considered consistent with Phelan-McDermid syndrome. And as I mentioned before, whether to report this CNV would be according to the local policy. For those participants who applied the American guidelines, again, the majority of participants classified the CNV as a VU, and there was a lot of consistency in the scoring using the guidelines. All participants correctly assigned a score of 0 to sections 1 and 3. Section 2 showed a bit more variability. Most participants applied 2J with a score of 0 and then applied 4D with a bit more variation of score depending on what literature and obviously their internal laboratory data showed. Again, limited inheritance pattern or family history information was provided, but nearly all participants applied a score of 0 for 5F. And again, we can see a lot of consensus over the classifications, with the vast majority of participants agreeing with the assessors and giving the expected classifications. So there was variation in whether this CNV would be reported. Less than half participants would definitely report this CNV. Just over a quarter would await the results of parental testing prior to reporting, and just over a quarter would not report this CNV. And as obviously the reporting of variants of uncertain significance is down to local policy, this aspect of case 2 was not marked. For case 3, this was a young boy who had intellectual disability, feet anomaly, an unspecified corneal anomaly, and his results showed a paternally inherited 276.5 kb loss of 8q22.2. However, there was no phenotypic information given for the father, and there was no further family history information given. The assessors expected that the CNV should be classified as pathogenic, and there were some issues with this, and the clinical significance would be a variant of uncertain significance for this particular patient. And because the CNV was classified as pathogenic, this should be reported to allow follow-up, which I'll go into in a minute. So the CNV itself is an intragenic loss of the VPS13B gene, which is a well-described OMIM morbid disease gene. However, it's associated with a recessive disorder, that of Cohen syndrome. So heterozygous losses are reported in unaffected individuals, but where there's a biallelic loss of function, this would be associated with Cohen syndrome. And because obviously of the presence of heterozygous losses, this comes up in the database of genomic variants as a polymorphic variant. In terms of our patient, as Angharad alluded to earlier, with recessive disorders, you really need to have a look at the patient's clinical features and see if they could be consistent with the recessive disorder in question. And it was considered that these clinical features were suggestive of Cohen syndrome in this patient. But obviously, the CNV alone cannot be considered to be causative of the patient's phenotype because there's no evidence of the second pathogenic variant at this stage. So therefore, further studies are required to clarify the clinical significance of this CNV. The assessors considered that as the CNV itself is classed as pathogenic, it should be reported to allow the appropriate follow-up. So applying the American guidelines there were a few issues with this. So although all participants correctly assigned a score of 0 to sections 1 and 3, section 2 scoring is complicated by the recessive nature of the gene. And we found that the majority of participants applied to E with the maximum score of 0.9, but there were several comments from participants that they were unable to score section 2 because of the design of the guidelines. And there was also wide variation in the scores assigned to section 5 because of the incomplete family history and phenotypic information. So we can see that there's much wider range of classifications were given for case 3. The assessors expected that the CNV should be classified as pathogenic, but nearly as many participants classified the CNV as a VU, possibly because of the issues with the American guidelines, but also whether they had been truly uncoupling the CNV from the clinical significance. I think you can see that virtually everybody agreed that the clinical significance was a VU or likely pathogenic. And in terms of reporting this CNV, about 2/3 of participants would report it, and just over a quarter would await parental results prior to reporting. However, there were 4.6% of participants who decided that they would not report this CNV, and this was marked as a critical error because the assessors considered that this should be followed up with further testing, and by not reporting the CNV, this could not take place. Just to mention this, although this is marked as a critical error, this is a pilot EQA, so there's no performance significance assigned to this. So as I mentioned, there were several challenges that case 3 demonstrates. So the American guidelines are intended primarily for the evaluation of CNVs associated with genes or genomic regions associated with dominant Mendelian inheritance patterns and don't necessarily apply to recessive disorders. There are also issues with applying the guidelines where several laboratories actually provided evidence for a pathogenic CNV classification, but in the end, they didn't categorize it as pathogenic. And this demonstrates the need to apply your own professional judgment to the clinical information and results obtained and not just rely on the score that comes out of a calculator or just by adding up the results on the table. And there's clear consensus that the disclosure of carrier status is recommended for well-described recessive disorders such as Cohen syndrome with clear clinical consequences. And as Angharad mentioned, the UK proposals to extend the ability to use the ACMG frameworks for classification of CNVs containing only recessive genes is in place, and there's a working group looking at extending that. So case 4 was a small girl with no speech and developmental delay. Her facial features were described as similar to Angelman syndrome, but the family history was currently unknown. And there were 3 CNVs given for this case. So the first one was a 794 kb loss of 1q44, which the assessors considered should be classified as likely benign and would be likely benign clinical significance for the patient and therefore should not be reported. There was a 1.37 megabase loss of 15q13.2 to q13.3, and this should have been classified as pathogenic with a pathogenic clinical significance and therefore should have been reported. The 121 kb loss of 17p 13.3 was more tricky, and the expected results were a likely pathogenic classification and a VU clinical significance. But because this CNV could be also contributing to the phenotype as well as the 15q, it was considered that this CNV should also be reported. So the justification for these classifications is that the 1q would be considered to be likely benign. It encompasses a cluster of olfactory receptor genes, but there's no known disease association associated with this region. And it would be likely benign rather than benign because the DGV data does not quite reach 1% frequency. The deletion would not explain the patient's phenotype and really should not be reported or followed up in this patient. Conversely, the 15q loss overlaps the 15q13.3 microdeletion syndrome region, and there's a lot of evidence that this would be haploinsufficient, and therefore this 15q CNV should be classified as pathogenic. The clinical significance was a bit more complicated. So although the patient's phenotype is non-specific, it could be considered consistent with the 15q13.3 microdeletion syndrome. But it is probably towards the more severe end of that, given that there was no speech and the level of developmental delay was not described. This CNV should be reported as it's pathogenic. Then the 17p loss. So this again was a bit tricky. The loss itself contains the YWHAE gene, which is associated with Melodica syndrome as part of a larger deletion. So the gene itself is thought to be a critical gene for this syndrome and is OMIM morbid dominant with lots of evidence for haploinsufficiency. But deletion of just this particular gene are rarely reported. And the clinical significance for the 17p deletion would be less certain. So it's possible that it's contributing to the patient's phenotype, but this CNV would not explain the absent speech, and so it's uncertain. But it's important because we can't distinguish which features are being caused by 17p or 15q. It should be reported to allow complete follow-up. So here are the bubble plots for the case 4. So quite surprisingly, there were a significant number of participants who classified the 1q CNV as a variant of uncertain significance, although there's a lot of evidence that for that to be classified as likely benign or benign. So again, that's possibly something to do with the application of the American guidelines and the scoring of the number of genes. The vast majority of participants assigned a score of 0 to section 1 and 3A should have been applied. For the 15q, again, this was less complicated with a lot of agreement amongst the participants. And for the 17p, as I said, again, this was a bit tricky. We can see that although the assessors expected the 17p to be classified as likely pathogenic, the majority of participants classified this as a VU. In terms of reporting these CNVs, again, it should be viewed as a whole for the case. So the 1q loss, 80% of participants would not report the CNV. But 15.2% of participants would report it and 4.5% would await further test results. So the assessors considered that in view of the case as a whole, this was not appropriate and a deduction was applied. There was lots of consensus for reporting the 15q loss with nearly all participants reporting that. And the 17p was a bit more variable, with about 3/4 of participants saying that they would report it and 13.6% of participants awaiting further test results. But 10% of participants would not report it, and here again a deduction was applied, as it's possible that this CNV is also contributing to the phenotype and it could have significance for other family members. So there were some challenges, obviously, again demonstrated by case 4. Section 2 should not be applied. Some participants applied a 2F with a score of -1, but there's section 2 should just not have been applied because there's no overlap with established or predicted haploinsufficient or benign genes or genomic regions. And as I mentioned just now, the scoring of the gene cluster also presented some challenges. When applying the American guidelines to the 17p loss, this highlighted some issues as well, because although the deletion fully encompasses the haploinsufficient gene, this is usually seen as loss of a larger 17p13.3 deletion in the patients with Mendelian syndrome. So it's uncertain as whether a 2A complete overlap of the gene should be assigned, or 2B referring to the genomic region. And we saw that most participants applied 2B and 2H, and then there was variable scoring for section 4, which resulted in this view classification that we saw from the majority of participants. So the general comments referring to this EQA was that the participants themselves showed a consistency in their approach to the EQA cases. So they either used their own justification for all 4 cases, or they applied the American guidelines to all 4 cases. However, as I mentioned, there were some exceptions for case 3. Due to the recessive nature of the condition involved, there were some laboratories who chose not to apply those guidelines for that particular case. And there were just less than half of participants who used the guidelines for these EQA cases. But obviously it's possible that others might use the guidelines on an ad hoc basis diagnostically, and particularly perhaps for those variants that are a bit more hard to classify, that fall into the uncertain region. As I've demonstrated, where participants did apply the guidelines to these cases, there was quite a lot of variability, particularly in cases 3 and 4. And whether the participants applied the guidelines or used their own justification influenced the performance, but this was different for each CNV. So it wasn't necessarily that those who always applied the guidelines always scored better. It just depended on the CNV. So the participants who used the guidelines came out with higher scores for cases 1 and 2, but they made more errors in case 3. And again, perhaps this is a reflection of the design of the guidelines as they are at the moment. And the participants who used their own justification scored higher for the 1Q CNV in case 4. And as I mentioned, that might be due to challenges in scoring the gene number. So that brings us to the end of the summary of the recent EQA. So looking forward to the future, we'll be running this EQA in a similar format again in the autumn, and we'll also at that time be offering a similar EQA focused on prenatal CNVs. These will both be online EQAs, so it's not too late to enrol in them. If you'd like to enrol, just email us or have a look on the website for any further information. We've also got an exciting module under development, which we call GTACT. So the EQAs that I've just described would be applicable to laboratories, whereas GTACT would be an individual competency assessment. And we're hoping this will be a really good tool for individuals to use for training and to assess ongoing competency. So we'll keep you updated with our software developments for that. And then looking forward to 2022, we are hoping to include CNV classification in a couple of our other EQAs, so the exome sequencing data interpretation EQA and the variant validation EQA. In 2022, they'll both include CNVs. So that concludes my part of the presentation. Thank you.
So I'd just like to say thank you very much to Angharad and Melody for two brilliant talks there. We have a few questions that hopefully Melody, Sean and Angharad will be able to answer for you. The first one actually is probably a mixture of all three of you, and it's to do with the classification of the YWHAE gene. We've got a couple of comments about the Clingen score for this being 0 rather than 3. Could you answer that one, Melody, maybe?
0 rather than 3. I think it's to do with the fact that it's rarely reported this deletion was only, I think it was 121 kb. So obviously the Mendelian syndrome patients have much larger deletions than that. I can look into that in more detail and reply individually to that question as well.
Okay, that sounds like a good idea. We've got a couple of other questions. One is, how should we deal with CNVs which include imprinted genes?
So I think these need to be interpreted in the context of the case. So if you look in the supplementary guidelines that accompany the ACMG, they do say that in following them that you should consider the parent of origin imprinting effect. So this means that some of the guidelines, similar to case 3 where the guidelines don't quite apply perfectly for autism or recessive disorders, it may be that you can't apply the guidelines in quite such a black and white format for imprinting disorders, and that you need to understand and apply the context of the disease, especially when you're looking at information such as inheritance studies and how it segregates through the family. So you might need to adapt them and use your judgment alongside using the guidelines as a start point.
Okay, thank you for that. We also have a question about if we are strictly applying the ACMG Clingen guidelines and detect something like the standard 22q1.2 recurrent microdeletion in a patient with consistent phenotypic features, but have no information on the inheritance pattern or family history, how would we score section 5?
I think for this section we would use 5H, and that the patient has the phenotypic features specific for the disorder, but that we haven't been given the family information. And that's how I would capture it using that code, I think.
Okay.
Yeah, I would agree.
We've also got a question, can you clarify the difference in taking part in this postnatal CNV classification scheme and the standard postnatal CNV scheme?
Yes, I can answer that. So the standard scheme that we've been running, which is an accredited EQA, involves us sending out DNA samples to you for you to process using your routine methods, and this could be by microarray testing or by sequencing. And for this EQA, you'd be expected to test the sample, analyse the results, and provide a fully interpreted report. The EQA that I've just summarised here is a purely online exercise where we provide the referral details and the results, and what you have to do is interpret them, putting your results onto a pro forma, so literally just classifying the CNVs, whether you think they're pathogenic or not, and providing a short summary of the justification for that. We're not marking your usual laboratory report, you're just filling in the pro forma, and we're gathering that data because everybody will obviously be given the same referral details and the same results, and we can compare participants directly to each other.
That's great, thank you, Melody. That's us kind of out of time now. We do have a few other questions, but we'll respond to those people individually through the website. So it just remains for me to say thank you very much to Angharad, Melody and Sian, and we hope to see you at the next Focus On session.
Thank you.
Yes, and also just to mention that if you did miss any of this webinar, it will be recorded. It will be available to view in the next couple of days for those of you who have registered, and it will be freely available for colleagues or anybody else who wants to view it on YouTube in about a month. If you have logged on today, you will receive a participation certificate automatically through the website and a feedback survey, and we'd be grateful for your feedback for this webinar. So that concludes the presentation. Thank you very much.